SAQs of immune system chapter. So today, let us go with some of the important SAQs that which are there in the genetics and reproduction also. In reproduction chapter and genetics, whatever the important SAQs are there, in today's session, we are going to learn them. And one more thing, if you have any queries, last 5 minutes, you will be unmuted so that you can clarify all your doubts. And even whatever the videos, recorded videos are there, they are getting uploaded in the YouTube. So you can visit the particular website, Social Data Initiative Forum, and you can watch the videos. Wherever you get the doubt, again and again you can watch. So today, let us go with short answer questions of reproduction. Reproduction means here specifically from the reproductive health. You will get an important SAQ, that is, why is sex education necessary in schools? So this is important SAQ. Why is sex education necessary in schools? Because in previous exam papers, this question has been given. So only this question from the reproduction chapter, if you learn, that is enough. So why is sex education necessary in schools? So sex education is obviously necessary in schools. And in order to give awareness about the sexual practices and the process of reproduction and the sexual practices, in order to give complete awareness about them, and in order to avoid the misconceptions, whatever are there related to reproduction or related to sexual practices or sexually transmitted diseases. So all the misconceptions and complete information and awareness will be given through the sex education in schools, because the adolescence is the age where it is with so many confusions, so many doubts, where your body is undergoing physically and mentally so many changes. So that's why, in order to give complete awareness and in order to have proper reproductive health care, so the process of this sex education is introduced in the schools. And many government and non-government agencies, they are working towards this reproductive health care program, and mainly by using different types of audio, visual and print media. By using audio, visual and print media, so mainly they are giving so much awareness related to this sex education. So what will happen when you learn about the sex education? So mainly in this, you are going to learn about the process of reproduction, about the reproductive organs, that is male reproductive system, female reproductive system, so that you will have a proper reproductive health care. And apart from that, even it gives the information about the contraceptive devices or the family planning program. So family planning program or the contraceptive devices, and even it gives the information about STDs. What is STDs? Sexually transmitted diseases. So it gives information about sexually transmitted diseases. So all this information is necessary in order to know and in order to avoid the misconceptions, in order to grow in a healthy way. So even in the reproductive manner also. And so that with this information, particularly what happens in the adolescents or the youth, so they will be careful and they will be trying to practice a proper or the safe reproductive health care. And not only that, even with the help of this sex education in schools, even it gives information about the care need to be taken for the pregnant mothers. So that means pregnant mothers, what kind of care, what kind of food they need to take, and what kind of exercises they need to do in order to have a safe delivery. And not only that, postnatal care. So that means both prenatal care and postnatal care. That means after the birth of the baby, even how the baby need to be fed with, and what is the importance of the breastfeeding. So all this will be learned with the help of this sex education, so that it helps in avoiding the mistakes whatever done. So during the process of this delivery or prenatal care or the postnatal care. So whatever the problems or the mistakes that which are arising can be avoided with the help of the information and proper awareness which is given about, the care need to be taken for the pregnant mothers and the postnatal care. That means after the birth of the baby, what kind of food need to be given and what is the importance of the breastfeeding and how the care need to be taken. Everything will be learned with the help of this sex education. And not only that, with the implementation of this family planning program, it helps in bringing about healthy families. It helps in bringing about healthy families of desired size. It helps in bringing about the healthy families of desired size. So what is the meaning of this? That is we too and for us too. So hum do, hamare do. So that means with the help of this, the population size can be controlled. So before, like previously, what used to happen? So everyone, they used to have some 5 child or 10 child or even 15 child also, if you see the olden days, right? So with the, because of that, what was happening? So the population size was increasing enormously, that which has led to again many problems like economically or socially, okay, or financially, like this, different kinds of problems was arising. So in order to avoid that, so this sex education will give the right information and even the problems that which will arise when the population size increases or in the particular family when more number of child are there, what are the problems that which arise. So everything, the proper information will be given so that they can understand. And not only that, even they will give information about the safe sexual practices or the use of the contraceptive devices, or with the implementation of this family planning program. So it helps in bringing about healthy families of desired size. And not only that, with the help of the sex education, even awareness will be given to the people that equal opportunities for both male and female, equal opportunities for both male and female, for both male and female, and even the respect and proper care towards female child, the respect and proper care towards female child. So all these will be given. So that means with the help of this sex education, even it makes us to understand that both girl child and boy child, both are one and the same. So there is both should be educated equally and both should be brought up equally. So there should not be any differentiation between male and female child, because both of them have so same kind of caliber. So it depends upon how they are using. So you know that actually if you see the olden days, only the male was given more importance and the female child was not, they were not allowed to go to schools, or they were given least importance. So they used to feel that female child is a negative one. And because of that, what was happening? Female feticides was happening. Yes or not? So what are female feticides? So that means during scanning, during pregnancy time, when they come to know that it is female child, so they use it to force the woman in order to undergo abortions. So that means killing the baby in the mother's womb itself is only called as what? Female feticide. So that means this sex education, even it gives the information and even it makes us to understand that female feticides are wrong, and it makes us to realize that so both should be treated equally. Boy child and girl child should be treated equally. And not only that, even it makes us to understand all these sex-related abuses, sex-related crimes. So everything can be avoided with the help of proper guidance and counseling. So this is the importance of the sex education in the school. So the reason for introducing sex education in the schools is because this is the right age in order to realize what is good, what is bad, and what is exactly right. So what is this reproductive health? Just like how we treat other organs or other parts of the body, in the same way, the reproductive organs are also, it's a part of the body only, where they need to be taken in a hygienic care and a safety need to be taken. Otherwise, what happens? So there are many diseases that which are related to this reproductive system also. So that's why mainly the government and non-government agencies, with the help of this audio, visual and print media, so they makes us to understand what are these reproductive organs and what is the functioning of them, And what are these contraceptive devices or family planning devices? With this, no, all unwanted pregnancies can be avoided. And with the avoiding of unwanted pregnancies, obviously the population size can be controlled. And even it makes us to understand about sexually transmitted diseases. If care, proper and safe sex is not being done, how many sexual transmitted diseases are going to affect the people. And even it gives information about the care which need to be given for the pregnant mothers, and even the postnatal care, and importance of the breastfeeding. So because the mother's milk, the first milk whatever been given, it contain IgA antibodies that which are very important for the baby, so to protect from various diseases. And even it helps in bringing about healthy families of desired size. And even it makes us to understand that equal opportunity should be given for both male and female child, and the respect and care towards the female child, and even in avoiding this female feticides. And even it helps us to understand about what are the different types of sex related abuses that which are happening, and sex related crimes which are happening, and how we need to be safe and make us to avoid them. So this is the importance of the sex education in school. So this is very easy. It's a common and general thing. So mainly you should know what should be written, like mainly female feticides, importance of breastfeeding, sexually transmitted diseases, and sex-related abuses or crimes. Such kind of terms when you remember, so easily you can make your own sentences and make the answer. So like this, very easily you can present this question, that is, why is sex education necessary in the schools? So only this question you can learn from this reproduction chapter. And next, coming to genetics. So coming to genetics, so let us see what are the important SAQs in genetics. So particularly in genetics, today I am going to teach 3 important SAQs. In that, first one is sex determination in case of human beings. How is sex determined in humans? That is important question. And salient features of Human Genome Project. So first one is, so genetics SAQs, sex determination in human beings. Sex determination in human beings. So humans have 23 pairs of chromosomes. So totally how many chromosomes are there in humans? So humans contain 23 pairs of chromosomes. In this 23 pairs of chromosomes, 22 pairs are autosomes. So 22 pairs of chromosomes are called as what? Autosomes. And the last one pair is only called as allosome or sex chromosomes. The last one pair can be called as what? Allosomes or sex chromosomes. So this is the first point which you should remember. So in human beings, totally 23 pairs of chromosomes are present. In this, 22 pairs are called as autosomes, and the last one pair is called as allosome or sex chromosome. So this autosomes will code for vegetative characters. So that means whatever the characters that which are there apart from reproduction, apart from the sex. So those characters are called as what? Vegetative characters. And whereas the chromosomes that which are responsible for determining the sex of the individual, that which is responsible for determining the sex of the individual, then it can be called as what? It is called as allosome or sex chromosome. And in case of males, so males are heterogametic. The last one pair of chromosome, that is allosomes in case of male. Allosomes in case of males are heterogametic. What is the meaning of heterogametic? So that means they are able to form 2 different types of gametes, like one gamete with X chromosome and another gamete with Y chromosome. So that means they contain XY. So that's why they are called as what? Heterogametic. So males are heterogametic containing XY chromosome. And whereas female, when it comes to the female, so females are homogametic. So that means they contain only one type of chromosome. So that is the same type of chromosome, which is X chromosome. So that's why males are called as heterogametic, and whereas females are called as homogametic. So what is the reason means? Because in case of the males during spermatogenesis, so that means during the production of the sperms. So during the production of the sperms, what happens? In this, whatever the sperms which are produced in male, 50% of the sperms, 50% of sperms will contain or carry X chromosome. 50% of the sperms will carry X chromosome, and whereas the remaining 50% of the sperms, they contain or carry Y chromosome. So during the process of the spermatogenesis, whatever the sperms which are produced in males, so in them 50% of the sperms contain X chromosome, and the remaining 50% of the sperms, they contain Y chromosome. And whereas in case of the female, so that means during this oogenesis process, so whatever the ova that which are produced, so during oogenesis, so whatever the eggs that which are produced, ova are the eggs that which are produced, all of them, they contain only one type. So that is X type. So that's why they are called as what? Homogametic. So that means with this, one can understand that now the sex of the baby or the sex of the child will be determined based upon the type of the gamete that which is getting fused with the female gamete. So that means the type of male gamete. Which type of male gamete is getting fused with the female gamete? So the sex of the baby can be determined. So it is very simple. So for example, if you take a male parent and female parent, male parent and female parent, as I told you, so whatever the sperms which are produced by the male, in that 50% of the sperms, they contain X chromosome, and 50% of the sperms, they contain Y chromosome. But whereas in case of the female, so the ova, whatever produced, they contain only one type of or same type of chromosome, that which can be called as what? X chromosome. Now, depending upon what type of gamete or what type of sperm is getting fused with the ovum, the sex of the baby can be determined. For example, here, if the fusion of the X chromosome of the sperm with the X chromosome of the ovum takes place, X chromosome of the sperm with the X chromosome of the ovum takes place, so then the sex will be XX. So that means XX will lead to female child or female baby. And whereas if the Y chromosome of the male fuses with the X chromosome of the female, so then it results in the production of male child. So like this, female child and male child. So with this, one can understand that depending upon the type of the sperm or the type of the gamete containing either X chromosome or Y chromosome getting fused with the X chromosome of the female, so the sex is determined. So with this, one can understand that, so what is responsible for the sex of the child? Y chromosome of the male helps in determining the Y chromosome of the male only helps in determining the sex of the baby. So because 50% of the sperms contain X chromosome and 50% contain Y. So here Y is only getting differed with. So that's why if the sperms containing Y chromosome are fusing with the ovum of ovum containing X chromosome in case of the female, so then the resultant will be what? XY, leading to the production of the male child. So with this, one can understand that the Y chromosome of the male helps in determining the sex of the baby. It helps in determining the sex of the child or the baby. So this is only called as sex determination in human beings, where you can present this answer like this in a simple way. So that is, first of all, human beings, they contain 23 pairs of chromosomes. In that, 22 pairs are called as autosomes, that which will code for vegetative characters. And the last one pair is called as allosome, or it can also be called as sex chromosome, which determines the sex of the individual. So here, In case of the male, always males are heterogametic. Why they are called as heterogametic? Because they contain one X chromosome and one Y chromosome. So X chromosome from mother and Y chromosome from father. And whereas female are homogametic, so containing same type of chromosomes. So that is X chromosome from father and even X chromosome from mother, leading to XX, which is female. So during spermatogenesis in case of the male, 50% of the sperms contain X chromosome and 50% of the sperms contain Y chromosome. And whereas in case of the ova, in case of the female, all the ova, they contain only one type, same type of chromosome, that is X chromosome. Now, depending upon what type of sperm is getting fusing with the ovum, that means whether the sperm containing X chromosome is getting fusing with the ovum or the Y chromosome is getting fusing with the ovum, the sex of the baby can be determined. If the X chromosome of the male is getting fused with the X of the female, so then the resultant will be XX, so which is female child. Whereas if the Y chromosome of the male, I mean that particular gamete is getting fused with the ovum, then the resultant will be XY, which is a male child. With this, one can understand that actually Y chromosome of the male helps in determining the sex of the baby. So this is about sex determination in case of human beings. So hope that you understood. If you have any doubts at the end of the class, so I'll give you 5 minutes so that you can ask. The next coming to very important question. So in the recent exam, whatever you have written, that this SAQ came, that is Erythroblastosis Foetalis. Yes or not? So this question has come. Erythroblastosis Foetalis. Actually, Erythroblastosis Foetalis, you can remember like a story. When you remember like a story, that will be very easy for you to present. So let us now discuss about Erythroblastosis Foetalis. Erythroblastosis Foetalis. So what is this Erythroblastosis Foetalis? So this problem arises actually whenever a RH positive father or RH positive male marries to a RH negative mother. RH negative mother. So whenever a RH positive father marries to a RH negative mother, and if the resultant child, whatever born, if it is RH positive, so that is not the problem during first delivery. So that means during the in the first child case, that is not at all a problem. Actually here what happens? RH, actually this mainly depends upon RH antigen. First of all, what is this RH antigen? So RH is an antigen that which is present on the RBC, red blood cell. So generally the red blood cell, it contains two different types of antigen. One is blood group antigen and the other one is RH antigen. So RH means rhesus factor. So rhesus factor. For example, now you will be telling that you will be saying like my blood group is A positive, or my blood group is O positive, or my blood group is B negative. So what is that positive, negative indicates? That positive or negative is depending upon the presence or absence of RH factor only. If the RH factor is present, then you will say a particular blood group as positive. So for example, if my blood group is A, so and even RH factor is also present on my red blood cells, then I say that my blood group is A positive. If RH factor is not there, then my blood group will be A negative. Like that, depending upon the type of the blood group and even the presence of the RH factor, so that becomes positive, or that becomes, if RH factor is not there, so that becomes negative with the particular blood group. So here blood group antigen may be A, or it may be B, or it may be AB, or otherwise if nothing is there, that will become what? O. O is actually 0, nothing is present. So O positive means what? Only RH factor will be there. O negative means related to the blood group and rhesus factor, no antigens are present at all on the RBC membrane, then that is negative, RH negative. So like this, depending upon the presence or absence of RH factor, we generally call it as RH positive and then RH negative. So whenever a RH positive father marries to RH negative mother, and if the first child is RH positive, what happens? During the delivery of the fetus, some amount of the fetal blood. So during this first delivery, some amount of fetal blood will be mixed with mother's blood. Some amount of fetal blood is mixed with what? Mother's blood. So whenever the fetal blood is mixed with mother's blood, now whatever the RBC which has entered into the mother's blood, as it contain RH positive antigen. So this is something like a foreign agent now to the mother, because mother is not having RH factor. So whatever which is not present, and if anything is coming newly into the body, that will be considered as a foreign one only, right? So that's why now mother's blood, as RH positive antigens are present on the RBC of the fetus, and such kind of RBC when they enter into mother's blood. So now what happens? So mother will become immunized and produce antibodies against to this RH positive antigen. But the first delivery will be safe. That means the first child will be safe. So the reason is it takes time for the mother to develop antibodies. And even the antibodies should cross through placenta and enter into the fetus. So that's why it takes minimum 24 hours time for the mother to develop the antibodies. But before the delivery has happened and the baby has been separated. So that's why the first child is safe. But what happens? Again, during second. So again, even if the second child is RH positive, even if second child is RH positive, so then what happens? Already now in the mother's blood, memory cells are also developed. Not only antibodies, even memory cells also have been developed. So now again, whenever the embryo is growing, and what happens now during this second pregnancy time, mother will become iso-immunized and still develops more and more memory cells, and thereby produce, that will differentiate into plasma cells and produce antibodies. Now these antibodies can easily pass through the placenta. It can easily pass through the placenta and enter into the fetus. Now the antibodies can pass through the placenta and enter into the fetus. So thereby what will happen? So this antibodies will bind to the RH positive antigen present on the, so RH positive antigen which is present. So thereby what will happen? So it results in agglutination. So that means clumping of blood. So the RBC will start to destroy. So clumping of blood will take place. So that means the RBC cells will start to die. As more and more RBC are getting destroyed, so the fetal liver will start to produce more and more RBC. Okay. Or even after birth also, the fetus will, the fetal bone marrow will start to produce more and more RBC. So that which can only be called as what? Erythroblastosis Foetalis. And such kind of babies, whatever born, so they are called as blue babies. The babies, whatever born, they are called as what? Blue babies, because most of the RBC are getting destroyed. So they develop anemia, and even they born with the jaundice. Like that, so many features can be seen. So again, I'm repeating, erythroblastosis foetalis is a condition where whenever a RH positive father marries to RH negative mother, and if they have RH positive child during their first pregnancy, and during delivery time, what happens? Some of the fetal blood will be mixed with the mother's blood, so that the fetal RBC containing RH positive antigen will sensitize the mother. Thereby, mother will develop antibodies against this antigen. But as it takes time for the antibodies to develop, and before that delivery has happened, the first child will be safe. But in the mother's blood, memory cells have been stored. Now what happens during second pregnancy? Again, in second pregnancy, if the baby is Rh positive, and mother will get isoimmunized and still produce more and more memory cells that which can differentiate and produce antibodies. Now these antibodies can easily pass through the placenta of the growing baby, okay, or the fetus. So now these antibodies can pass through the placenta and enter into the fetus, thereby destroying fetal RBC by binding with Rh antigen. So antigen antibody complex are formed. So thereby it leads to agglutination or clumping of blood or destroying more and more RBC. So because of that, what happens? So more and more RBC can be produced either from fetal liver or after birth from the fetal, after birth from the child bone marrow. So which is only called as erythroblastosis foetalis. And the babies who ever born can be called as blue babies. So sometimes what happens if major destruction of this RBC occurs, even there is chance that the baby may die in the mother's womb even before delivery. So like that, depending upon the condition, anything may happen. So which is only called as what? Erythroblastosis foetalis. Then what should be done? So if they come to know that generally by blood grouping, when the mother is pregnant, blood grouping will be done, right? So if they come to know that father's blood is Rh positive and mother's blood is Rh negative, so that's why now during like checkup and all, what they will do. So particularly in case of the second pregnancy, so it will be administered with Rogam antibodies or anti-Rh antibodies. So whenever anti-Rh antibodies or Rogam injection is administered, so then what will happen? These antibodies will be neutralized. Whenever the antibodies are neutralized, so that antibodies will not cross through the placenta and it won't destroy the fetal RBC. Like that, the fetus can be saved and even the mother also can be protected. Particularly the fetus can be saved. So this is only called as what? Erythroblastosis foetalis. So hope that you have understood. Then next, coming to let us discuss one more SAQ, that is Human Genome Project. Human Genome Project. So salient features of HGP, Human Genome Project, which is very important question. So this Human Genome Project, salient features, at least a few features you should remember and write, many are there. So it has been done. So that means this project has been started in order to assess the entire genome which is present in the human beings. So when they have done this Human Genome Project, let us see what are the main things which they have found out. So salient features of the Human Genome Project are, with the help of this Human Genome Project, it has been found that human genome contains nearly 3164.7 million nucleotides. It contains nearly 3164.7 million nucleotides. So definitely here numbers you should remember. So the human genome contains 3164.7 million number of nucleotides are present in the entire human genome. So that means in all the, like if you take a haploid set of chromosomes, that is 23 chromosomes. So in all these chromosomes, the number of nucleotides that which are present are 3164.7 million number of nucleotides are present. And the human genome contain approximately 30,000 genes. So approximately how many genes are present? 30,000 genes are present. In this 30,000 genes, nearly 50% of the genes functions are unknown. Even though 30,000 genes have been discovered. In this, 50% of the gene functions are unknown. 50% of the gene functions are unknown. And less than 2% of the genes are coding for the proteins. So less than 2% of the genome, are coding for proteins. So that means less than 2% of the, if you take the entire genome, in that entire genome, less than 2% of it is only coding for characters. You know that generally whenever a gene gets expressed, so the expression of the gene comes out in the form of a character. So coming out in the form of a character means what? Coming out in the form of a protein. So already you would have learned in molecular biology that, so a part of the DNA or a segment of the DNA that which codes for a character is only called as gene. So whenever the gene get expressed, the information present on the DNA or the gene is transcribed into mRNA. And again, the information present on mRNA is decoded into protein. That means expression or the characters are coming out in the form of the proteins. So less than 2% of the genome are coding for proteins, and 50% of the gene functions are unknown. And human genome contains 3,164.7 million number of nucleotides, where approximately this entire genome consists of how many genes? 30,000 genes. And again, each gene on an average may contain nearly 3,000 bases. So each gene may contain on an average 3,000 bases. That means almost in all the genes, at least 3,000 bases are present. 3,000 nucleotides or 3,000 nitrogenous bases are present. But if you consider the largest gene, so the largest gene is dystrophin gene. The largest gene is dystrophin gene, that which codes for dystrophin protein. So that which is required for the muscle. So the largest gene is a dystrophin gene containing 2.4 million bases. So hope that you have understood why it is called as the largest gene, because it is containing 2.4 million bases are present. Actually, every gene almost on an average contain 3,000 bases except the largest gene. So the largest gene is what? Dystrophin gene containing 2.4 million bases. And not only that, so most of the places in the genome, or most genes in the genome, or most places in the genome, actually they are differing only with respect to one nucleotide. So they are differ with one nucleotide. So majority of the places in the genome, so they differ with respect to one nucleotide. That means what? The remaining sequences are same. For example, imagine that in the genome, in this place, if there is a sequence like AGTC, AGTC. So here also, in the other place also, what happens? Same sequence is there, but only one nucleotide may differ. For example, like AGTC, AGCC may be present. In the same way, in some other place, ATTC, again AGTC. So that means they are differing only with respect to what? One nucleotide. So that is only called as what? SNP. Such kind of regions are only called as SNP. SNP means what? Single nucleotide polymorphism. Single nucleotide polymorphism. SNPs. They are called as SNPs. Single nucleotide polymorphism. And such kind of regions in case of the genome, that which are differing only with respect to one nucleotide. So almost how many locations like that are found means, so 1.4 million locations are found that which are differing only with respect to one nucleotide. That which are differing only with respect to one nucleotide. So which is only called as what? SNPs. Single nucleotide polymorphism. So they are differing only with respect to one nucleotide. So like that, how many locations have been found means 1.4 million locations have been found which are differing with one nucleotide. And again, if you consider the chromosomes with the largest number of genes and chromosome with small or shortest number of the genes or lower number of the genes. So the largest, the first chromosome, if you consider the chromosome 1 contain highest number of genes, that is 2968 genes are present. So chromosome 1, the first chromosome. So totally, if you consider 23 chromosomes in that, the chromosome 1 or the first chromosome contain 2968 genes. And whereas the chromosome Y, that means the 23rd chromosome. So chromosome Y, it is the shortest chromosome. So why it is called the shortest chromosome means because it contain only 231 genes. So the chromosome, first chromosome contain more number of genes. So that is 2968 genes. And whereas the Y chromosome, the shortest chromosome with the 231 to 241 number of genes are only present. So like this, the salient features of Human Genome Project. So human genome contain 3164.7 million number of nucleotides, where approximately it consists of 30,000 genes, and 50% of the gene functions are unknown, and less than 2% of the genome are only coding for proteins, and each gene contain nearly 3,000 bases. But the largest gene, dystrophin gene, is the one that which contain 2.4 million number of bases. And most places in the genome, they are differing only with respect to one nucleotide, whereas the remaining sequence will be similar. Such kind of locations are only called as SNPs, single nucleotide polymorphism, and 1.4 million such kind of SNPs are found out in the genome. So that is 1.4 million locations been found. And chromosome 1, it contains 2968 number of genes, and whereas Y chromosome is the shortest one, that which contain only 231 number of genes. So like this, at least some of the important salient features you should write about human genome. So in today's session, mainly 4 SAQs we have discussed. All these 4 SAQs today only you should learn. So what are that 4 SAQs? The first one is, how is sex education important? Why it is introduced in schools and colleges? So what is the reason for that? So mainly there, at least to remember some of the important terms, like what government and non-government agencies, how they are bringing awareness to people about sex education, particularly the adolescents. Okay. So mainly what is the importance of the breastfeeding, and what is the reproduction, then family planning program, then the prenatal care, postnatal care to be taken, and why the population size need to be controlled. Okay. So sex related crimes, sex related abuses, and equal opportunities given for both male and female child. So such kind of terms, when you remember easily, you can make the answer. And apart from that, we have discussed about sex determination in human beings. There you should remember males are heterogametic and females are homogametic. 50% of the sperms contain X chromosome, 50% contain Y chromosome. But in females, all of them, all the gametes, they contain only one type of chromosome, that is X chromosome. So depending upon what type of sperm is fusing with the ovum, so the sex of the baby can be determined. So mainly the Y chromosome of the male will become responsible for determining the sex of the child. And next is Erythroblastosis Foetalis, Rh positive father, Rh negative mother. When the baby is Rh positive, the first child will be safe, but mother's blood got sensitized with the development of memory cells and antibodies. And whereas in case of second pregnancy, again, if the child is Rh positive, now this antibodies, whatever been developed already, they will be passing through the placenta and enter into the fetus, where they destroy fetal RBC. So that what happens? So the fetal liver or the bone marrow will start to produce more and more RBC. So which is only called as erythroblastosis. And the babies born are called as what? Blue babies. And finally, the salient features of the Human Genome Project, where you should remember how many nucleotides are there, how many genes are there, and how many locations are SNPs, and the largest gene contain how many bases, and chromosome 1 has how many number of genes, Y chromosome, shortest chromosome, has how many number of genes, okay, and how much genome is coding for protein, and how much gene functions are still unknown. Like this, different points you should remember and write. So now it's time for clarification of your doubts. If you have any doubts, you can raise your hand so that they will be unmuted. Any doubts? So almost from the last 3 days, we are covering all the important SAQs. And once we complete SAQs, we will go for VSAQs. All the important VSAQs will be taught. Yes, any doubts? If you have any doubts, please raise your hand. So remember that definitely one SAQ will be given from digestion and absorption chapter. So definitely learn all the important SAQs from digestion and absorption, and even breathing and exchange of gases. Definitely one SAQ will be given. Then endocrine system, one SAQ. So if you learn like that, no chapters, you see now itself three SAQs you will be right when you cover all the important questions from digestion and absorption, breathing and exchange of gases, then endocrine system and chemical coordination. And definitely from genetics, one SAQ will be given. From evolution, one SAQ will be given. In tomorrow's session, we are going to learn important SAQs of evolution chapter. So here itself easily you can cover 5 SAQs. Yes, any doubts? Fine, if you don't have any doubts. So no problem, today you can check, and tomorrow when you join, you can come up with your doubts. So hope that you have understood. So thank you.